要试一下声音吗？可以。The title of my talk today is Defending the Importance of Lineage-Forming Reproduction in Evolution by Natural Selection. The conditions for evolution by natural selection are one of the most central and controversial topics in evolutionary biology and philosophy of biology. The evolutionary biologist and geneticist Richard Lewontin has provided a very influential framework to analyze the conditions of yin and S, in which variation, heredity, and differential fitness are proposed as sufficient as well as necessary conditions for yin and S. Although the sufficiency of these three conditions to produce yin and S has been convincingly challenged, people generally agree that they provide necessary conditions for yin and S to occur. The notion of reproduction is not explicitly mentioned in Richard Lewontin's three conditions. However, it still plays a very important role in his account. For example, the notion of heredity is understood in terms of the resemblance between the parent and its offspring. And the fitness of different variants is compared according to the actual or expected number of offspring it produces. The importance of reproduction has also been emphasized by many other biologists and philosophers. For example, Marcus Ridley, in his textbook for evolution, explicitly lists reproduction as a necessary condition for yin and S, besides the three conditions mentioned by Lewontin. Peter Godfrey Smith claimed that reproduction is at the center of Darwinism, because all discussions about heredity and fitness should be understood in terms of the parent-offspring relationship between individuals. Richard Dawkins and David Howe provide a somewhat different framework to analyze yin and S, in which they think that in order for a population to undergo evolution by natural selection, it needs to have both a replicator and an interactor. A replicator is an entity that can replicate itself. Here, replication is understood as a special form of reproduction. So reproduction still plays a very important role in Dawkins and Howe's framework. Although the notion of reproduction has played a very important role in traditional accounts of yin and S for a long time, recently a number of philosophers of biology have tried to challenge the necessity of lineage-forming reproduction for yin and S. For example, Bouchard argues that yin and S is to be understood in terms of ensembles and their parts, rather than populations and reproducing individuals. Biological systems like clonal species are ensembles that can undergo yin and S by differential persistence of their components without reproduction like genuine individuals in a population. Pierre Bouchard argued that reproduction should be understood as a product of yin and S rather than its necessary condition. Charbonneau and Papard argue that yin and S is a population-level phenomenon. So what really matters for yin and S is memory and regeneration at the population level. Reproduction with lineage formation on the individual level is not necessary for the occurrence of yin and S. In this talk, I will focus on Charbonneau and Papard's account of yin and S and try to defend the importance of lineage-forming reproduction in yin and S. So why do Charbonneau and Papard's accounts of yin and S deserve special attention? To answer this question, we need to distinguish between two types of yin and S, that is, paradigmatic ENS, and weak or minimal ENS. Paradigmatic ENS can produce complex cumulative adaptations, while weak or minimal ENS can only lead to distributional change in a population. It cannot lead to complex cumulative adaptations like our eyes. Most people may concede that reproduction is not necessary for the occurrence of weak ENS, but most of them will agree that paradigmatic ENS requires the existence of reproduction. By contrast, Charbonneau and Papin's account of ENS suggests that reproduction with lineage formation is not necessary even for paradigmatic ENS. So this demonstrates a more radical deviation from traditional accounts of ENS. So in this talk, I will focus on Charbonneau and Papin's accounts and defend the importance of reproduction with lineage formation in paradigmatic ENS. First, let me introduce Charbonneau's account first. Charbonneau understands reproduction in terms of multiplication plus inheritance. Multiplication is defined as any process where one or many entities or processes produce or participate in the production of a new entity of the relevant kind. An inheritance is any process through which an entity which is a parent affects another entity which is the offspring in such a way that the offspring resembles its parents with regard to the transmitted trait. Charbonneau also defines two other processes, but on the population level. One is called memory. Memory is a process of transgenerational retention of change in the distribution of variation of a population system. And the other is called generation, which refers to the renewal of population parts. Based on these concepts, Charbonneau argued that since ENS is a population-level phenomena, what really matters is generation and memory on the population level. Reproduction, which consists of multiplication and inheritance, is merely one of many possible ways to realize generation and memory on the population level. So it should not be regarded as a necessary condition for ENS to occur. Papin accepts the general approach of Charbonneau, but he makes some modifications. First, he redefines reproduction as any causal process of multiplication by which an individual has a privileged causal input on the structure, function, or material constitution of one or many individuals in subsequent generalizations. By privileged causal input, he means that a parent-offspring relationship can be drawn between individuals of successive generations, such that the causal influence of the parent is bigger and more direct compared with other unrelated individuals. Second, he emphasizes that ENS is a heterogeneous process, and he further distinguished between two types of ENS. The first is minimal ENS, the second is paradigmatic ENS. The occurrence of minimal ENS requires two necessary conditions. First is variation among individuals that leads to differential fitness in a population. Second is memory, which refers to the transgenerational rotation of change of the distribution of the variation distribution in a population with respect to some trait. And paradigmatic ENS requires three conditions, including A, B, and regeneration. Regeneration is a term used by Papert, but it is similar to Chaboneau's notion of generation. Okay, now here is the brief summary of Chaboneau and Papert's view. Basically, they argue that yin-s is a population-level phenomenon. Reproduction with lineage formation on the local level is not necessary for the occurrence of yin-s, because it is merely one of many possible ways to realize generation and the memory on the population level. Besides this conceptual analysis of yin-s, Chaboneau and Papert have also used various empirical cases to support their view. In this section, I will reconsider three such alleged cases in biology. It turns out that none of them can be used as evidence to demonstrate the existence of paradigmatic yin-s without lineage formation. The first case is about prions. Prions are misfolded infectious proteins. They can induce normal proteins to adopt their misfolded conformation and become new prions. Chaboneau uses the case of prions to argue that it is possible for a population to undergo evolution by natural selection without forming parent-offspring lineages when multiplying its components. And his argument can be reconstructed as follows. First, prions come from normal proteins. They have the same material base as normal proteins, but are misfolded in structure. Second, normal proteins are products of mRNA translation. In this process, neither pre-existing normal proteins nor prions participate in the material production of new proteins. Given 1 and 2, we get to the conclusion that no parent-offspring lineages are formed during the multiplication of prions. Different strains of prions can compete against each other, leading to a process of Darwinian evolution, that is, evolution by natural selection. Thus, a population of prions can evolve through natural selection, even though no parent-offspring lineages are formed during the multiplication of its components. We think that one problem with Chaboneau's argument is that he conflicts the notion of the multiplication of normal proteins with the multiplication of prions. Prions come from normal proteins, but they belong to different kinds of entities. When normal proteins are multiplied through the process of mRNA translation, prions have not been multiplied yet. Only when normal proteins are converted by pre-existing prions into the prion conformation can we declare the multiplication of prions. In this transformation process, pre-existing prions do play a distinctive causal role. That is, it induces the normal proteins to adopt its misfolded conformation. And this causal inference helps establish a parent-offspring lineage between pre-existing prions and newly produced prions. So there is a process of lineage formation here. Here's one possible reply from Chaboneau. He may argue that, well, this is not a case of lineage formation during the process of multiplication, because what pre-existing prions really do is to participate in the inheritance process, not the multiplication process. I think this view is faced with two further problems. First, it is unclear why material contribution should be regarded as a necessary condition for an entity to participate in a multiplication process. Because if we accept this view, then even mRNA cannot be said to participate in the production of new proteins, because mRNAs do not materially contribute to the production of new proteins. Second, even if we agree with Charbonneau that pre-existing prions participate in the inheritance process, not the multiplication process, a parent-offspring lineage is still established here. Since for Charbonneau, the notion of reproduction consists of both inheritance and multiplication. If there is a lineage established in the inheritance process, this lineage should also be established in the process of reproduction. Papin is not satisfied with Charbonneau's case of prions either. He thinks that holobions provide a better case for their argument. A holobiont is a functional entity composed of a host and all its symbiotic microbes. A host within a holobiont can acquire its microbes in two ways, that is, vertical transmission and horizontal transmission. In vertical transmission, a host acquires its microbes directly from its parents. In horizontal transmission, a host acquires its microbes not directly from its parents, but from a microbial source pool in the environment, which can contain microbes coming from anywhere. Papin appeals to the biologist John Roughgarden's model of horizontal microbiome transmission to support his argument. Roughgarden's model consists of three stages. The first is called microbial transmission. In this process, hosts from a host source pool and the microbes from a microbial source pool are sampled and combined together to form a new generation of holobionts. The second stage is called microbial proliferation. In this process, the microbes within each holobiont will increase according to certain kind of ecological model. The third stage is called holobiont selection. In this process, each holobiont as a whole will reproduce, and the hosts and the microbes in the new generation of holobionts will form a new host source pool and a new microbial source pool. Then a new cycle can restart based on these two source pools. According to this model, Papin argued that holobionts provide examples of regeneration without lineage formation. Because the population of holobionts is being regenerated by the random association of holobiont parts, that is, the hosts and the microbes, coming from two source pools. And this process is too diffuse to invoke lineage formation. For example, if we consider the microbes in a single holobiont, then these microbes can come from many different holobionts in the previous generation. So it's difficult to find a kind of parent-offspring relationship between different holobionts. Well, our response is like this. We agree that there's no lineage formation between different holobionts here, but we think that what is really relevant here is whether there is evolution by natural selection at the level of holobionts. In other words, we need to consider this question. Do holobionts largely rely on horizontal transformation count as units of selection? The criteria for units of selection are widely discussed in the literature, but still remain controversial. Nonetheless, we think there seems to be some minimal criteria that are widely accepted by philosophers and biologists in the literature. One of such criteria is called fitness alignment. Roughly speaking, in order to test whether a biological system counts as a unit of selection, we can manipulate a character of a unit within this system and see whether it would will affect the fitness of all the partners of this unit with the same or very similar magnitude and in the same direction. Given this criteria, let's consider a fatal mutation appearing in the host of the holobiont. This mutation will kill the host and reduces its fitness to 0. The fitness of the microbes within this holobiont will also influence in a negative way, but with a very different magnitude compared with that of the host, because those microbes can live in the environment freely by themselves. Given this, we think that holobionts with largely rely on horizontal transformation do not really count as units of selection. So it cannot be used as an example to demonstrate the existence of paradigmatic yin-s without lineage-forming reproduction. The third case is called ecological inheritance. Charbonneau starts with an imaginary example. Suppose there's a population of bacteria in the lab, and there exist 2 types of variants in this population. The researchers introduce a kind of toxic chemical compound into this population, and one variant is more tolerant to this chemical than the other variant. So this difference in their tolerance to this chemical will lead to a process of natural selection. After a period of time, the researchers will record the information of variation distribution in population B and save the information in the computer. Then the researchers artificially synthesize a new population of bacteria according to the information of variation distribution saved in the computer. Charbonneau argued that in this case, memory is realized from population B to population C because they have the same variation distribution. However, there is no lineage forming between individual bacteria in B and C because we cannot find the parent-offspring relationship between individuals in population B and the individuals in population C. However, we think that this example is very problematic because there's no evolutionary relationship between population B and population C at all in this case. So it cannot be used as an example to demonstrate the existence of paradigmatic yin-s without lineage-forming reproduction. Charbonneau also considers a more realistic example. Females and males of different species of fish can release different chemicals called steroids into the water. If their offspring live in the same environment as the previous population, then the steroid level in the water can be transmitted to the next generation through the process of ecological inheritance. And this steroid level can further determine the sex ratio of the next generation. Charbonneau argued that in this case, there is memory on the population level with respect to the sex ratio of the fish population. However, there is no inheritance lineage on the individual level because the sex of each individual fish is determined by the steroid level in the water, which is further determined by all the fishes in the previous generation. So you cannot identify this kind of one-to-one lineage relationship between individuals in the offspring generation and the individuals in the parent of generation. We think that there are two problems with Charbonneau's argument. First, even if there is no inheritance lineage with respect to the sex of each individual fish, there is still multiplication lineage because each individual fish in the later generation is reproduced by its parents in the previous generation. Second, merely focusing on ecological inheritance leaves unexplained why the steroid level in the water can determine the sex ratio of the next generation. To answer this question, the key is to realize that individual fish in the next generation has inherited the capacity of developing a certain kind of sex according to the steroid level, and this capacity is inherited through the process of reproduction. And this reproduction process not only forms a multiplication lineage, but also an inheritance lineage. In other words, the example regarding the sex ratio of fish is perfectly compatible with the standard account of E&S. Until now, we have shown that at least in the realm of biological evolution, we have yet to find empirical cases of paradigmatic E&S without lineage-forming reproduction. Here is a possible objection from Charbonneau and Papard. They may argue that although we have yet to find empirical cases of paradigmatic E&S without lineage-forming reproduction, this does not mean that such cases cannot be found in the future. For example, Papard said that the proposed reformulation of Lewontin 3 conditions has shifted the burden of proof on those claiming a priori that populations of non-reproducers cannot sustain E&S. However, who really has the burden of proof? Our first reply is that, well, although it is true that we cannot claim a priori that populations of non-reproducers cannot sustain E&S, this merely shows that E&S without lineage-forming reproduction is logically possible, not that it is biologically possible. And merely logical possibility provides very little justification for advocating a population-level reproduction-independent reformulation of Lewontin's 3 conditions. Because in order to make a conceptual change in biology, one really needs to develop a case for the necessity of making such a change. Our second and presumably more substantive reply is that if we adopt Charbonneau and Papard's reproduction-independent account of E&S, we would actually confront a series of problems with respect to E&S. In the following section, I will talk about these possible problems. The first problem is that if we adopt their account of ENS, we will include some obviously artificial cases as genuine cases of ENS. So let's reconsider the imaginary example of the bacteria in the lab. In this case, it seems that both the conditions of regeneration and memory are satisfied. The memory is satisfied because population B and C have the same variation distribution. And the regeneration is realized because new bacteria are synthesized in population C, which can be understood as the renewal of population parts. However, as I have argued, population B and population C have no evolutionary relationship at all. So this should not be regarded as a genuine case of ENS. The second problem is that adopting their account of ENS will exclude some genuine cases of ENS. Let's consider such a population in which there are two types of variants. These two types of variants have the same survival ability, but they have different reproductive abilities. This difference in their reproductive abilities will lead to a genuine case of ENS. However, according to Charbonneau and Papar's account, this should not be regarded as a paradigmatic case of ENS because memory is not realized in this case. Remember that for them, memory refers to the correlation between successive states of a population with respect to the distribution of variation of a certain trait. And the more similar the successive states are, the stronger the memory is. And it's clear that the variation distribution between these two successive states, as shown in the picture, is very different from each other. So according to their standard, there's a very low degree of memory realized in this case. So it cannot be regarded as a genuine case of ENS. However, this contradicts with our common view that this is a genuine case of ENS. And this analysis actually showed that memory should not be regarded as a necessary condition for ENS. The third problem is about the cost of having too much abstraction. Darwin's original characterization of the conditions for ENS is full of details beside the three conditions mentioned by Lewontin. So in other words, we can say that Lewontin's formulation of ENS is more abstract than Darwin. However, we think that there should be some limitations on how abstract such a formulation could be. In Lewontin's formulation, when we take reproduction into consideration, it allows biologists to make some predictions about the evolution of population or biological population, although usually these predictions can only be made very qualitatively and very roughly. And in many cases, these predictions may fail. Nevertheless, they allow us to make some predictions. This is because when we are faced with a biological population, we can identify whether there is variation in this population, and we can check whether those biological entities can reproduce or not. And also, we are pretty sure that the offspring of these biological entities will tend to be similar to their parents, because as Darwin has already realized, inheritance is usually related to reproduction. However, if we merely focus on population level regeneration and memory, we are not allowed to make such predictions, because when we are faced with a biological population, we do not know whether the requirements of memory will be realized in the future. Okay, now we come to the conclusion part. In this talk, I have shown that Charbonneau and Papard argue that ENS is a population level phenomenon. Reproduction with lineage formation on the local level is not necessary for the occurrence of ENS. However, our empirical analysis showed that none of the cases cited by them can be used as evidence to support their view. And our conceptual analysis showed that adopting their reproduction-independent account of ENS would actually lead to many problems. Given this, we argue that the proposal of merely focusing on regeneration and memory on the population level when formulating ENS is unjustified. At least in the realm of biological evolution, lineage-forming reproduction is still necessary for a population to undergo paradigmatic ENS. Thanks.